Thank you very much. Thank you. We're making a concerted effort to actually start on time this year. So hello, I am Natural History Museum Exhibit 237A, Quentin Cooper, tweeting as @QWERTY, cross-tweeting as @Gertie, and your host for this Hall of FameLab as part of Science Uncovered 2017. If you're expecting anything else, get out now. A particular welcome to the people who are watching the live stream of this. This is being live streamed to over 200 countries, basically anywhere they have the internet, which is everywhere, really. So, as I'm sure you're aware, FameLab is an anagram of Fab Male, which is me, and Science Uncovered is an anagram of Denounce Crevices, which is irrelevant. But more relevantly, Science Uncovered is also an anagram of concise, even, crude, which is a pretty good way of describing what FameLab is about. Concise, because everybody has just 3 minutes. 3 minutes only, not a second longer, otherwise they hear, which is pretty damn scary as well, to get across something about the magnificence and significance of science, the wow and the how, the wonder and the thunder. Even because they have to strike a perfect balance between being informing and being entertaining, and crude because this is a slightly later night event than FameLab usually is, so we can be a bit more riskier and riskayer. Although this is still proper science communication, so don't expect a shitload of gratuitous swearing. So, for anyone who's new to this, are there any FameLab versions in? I stress FameLab versions, not just your regular versions. There'll be far too many hands. It's confusion now. Is it FameLab versions or others? And what's the Venn diagram with buttons? No, it's fine. Okay, so then congratulations. You have stumbled upon the world's largest and most fragrant science communication competition. It started at the Cheltenham Science Festival, and with the help of the British Council, it has spread to become a titan, a gigantic, a behemoth. It is in over several dozen countries on every continent apart from Antarctica. It involves top organizations like NASA and CERN and people from presidents and kings and queens to Lieutenant Uhura from Star Trek. I am not making any of that up. Now, the international final this year in Cheltenham had 31 countries taking part, and it went on for days. Tonight is a mere morsel, a taster, a bite-sized, fun-sized FameLab featuring 7 proven FameLab talents from all around the planet, or more accurately, Europe. We've got finalists from Bulgaria, Greece, Ireland, the Netherlands, Poland, Spain, and some rogue, politically unstable state called the UK. Each will strut their funky stuff for exactly 3 minutes, then face a couple of minutes questions from our 3 judges. And who are these 3 foolhardy judges, I hear you ask? That's good. I like an audience that knows a cue. So, thank you. Can we please welcome to the stage science communicator, physics outreach officer for Royal Holloway, University of London, and former FameLab FameLab Greece finalist, Anna Christofoulou. Next, as the seas get higher, so does the media profile of our second judge. He's a principal teaching fellow at the National Oceanography Centre at Southampton University. He's, like me, been a part of the ongoing science, art, climate change project Cape Farewell, and he's the ultramarine man who puts the current into current affairs, as pretty much any news story involving any aspect of climate or oceans or anything slightly wet he's the guy who turns up and is interviewed. Dr. Simon Boxall. Oh, him you're now going as he takes his seat, right. And final member of our triumvirate was, until about this time last year, Director of Science at the British Council, the organisation without which none of this would be possible, but is now Senior Director, Programmes at Explorer Development Europe, catchy title, for the National Geographic Society, an organisation without which this is entirely possible, but, you know, they're welcome to join in if they'd like to. Please welcome Dr. Claire McNulty. So Anna, first of all, welcome back. You were a judge last year. What drew you back to do this again? Most people make sure this is a once-in-a-lifetime experience.
Oh, I hope you won't get rid of me so easily. It's really nice. Everything is nice, and you're very good at this, Quentin, so just couldn't
That is
help.
absolutely true, and I need that kind of affirmation on a minute-by-minute basis. That's fine. So now we mentioned this is the 10th anniversary of FameLab International, and you took part in one of the very first FameLab International, and certainly the first one in Greece 10 years ago. Take us back. How has it evolved? Would you stand a chance in the competition now?
Oh, so this is a very hard thing to do, stand up here and try to dazzle the audience. So I'm very happy I'm hiding here at the judges' table for the moment. It has evolved greatly, hasn't it? I mean, so many countries. When we started, it was in the UK, started in 2005, and then it spread in 10 countries in the Southeast Europe in 2007. And now it's 31, and we have CERN, we have NASA. I'm very thrilled, and I just can't wait to see the next 10 years what's going to happen.
Yeah, this is far faster than the World. The World Cup took 48 years to get to the size that FameLab has got to in just 10. Fact, fans. Now, Simon, have you been surprised by becoming TV's favourite go-to guy for all things aquatic?
I'm the only person who'll actually get up at 5 o'clock in the morning and stand on a beach in the freezing cold. I think that's the main reason.
Right, you're just available.
I'm just available, yes.
But come on, there must be something about you that floats the boat of media folk.
I've no idea.
I'm puzzled myself. That's why.
I guess it's just I enjoyed. I mean, the oceans are a fantastic place. It's full of interesting science, and it's just sort of. You know, I just love the science of the oceans, so it's trying to get that across to the general public.
And that is not coincidental, because this year you've been selected as judge because we have a themed FameLab, fitting the general theme, which is
basically 72% of the planet. So I mean,
yes,
you know,
it's a big
at long
theme,
last,
isn't it?
it's a big theme. Yes.
Yes, so much as though we used to talk about the 7 seas, we've got 7 FameLabbers who are each going to do something on a marine theme, but that can be almost
any of those seas. Yes, absolutely.
Claire. We're sad to lose you, the British Council, but congratulations on it. Tell us a tiny bit, without it turning into a massive plug for National Geographic, about the new job.
Well, National Geographic supports scientists, explorers and storytellers, so it's very closely aligned to FameLab, I think. And I'm not going to tell you loads, but as a quick plug, we do have a call coming out shortly on oceans as well, so it's perfect timing. People in the UK are able to apply.
But when you say you support explorers, there's loads of people in this room who are probably going to say, well, I'm an explorer, sort of. So how would you support them? Go, here's a plane ticket, off you go to deepest Bolivia?
Well, if they've got an interesting enough project, yes.
Brilliant. You're going to be mobbed during the interval. And all the years you've done FameLab, of course, because you were involved with British Council, you've never been able to be a judge. No. Are we about to see some hideous partisan streak come out? Or
no, I don't think so. I think I was working out, I've seen about 12 FameLabs,
right.
International and national ones, but I've never judged, so I am actually quite scared. It's a big responsibility. So, yeah, we'll see.
Yeah, I've never done it either as well. And just remind us, particularly for those who are the virgins, what are the criteria you'll principally be judging our FameLabbers on tonight?
So we're looking for content, clarity and charisma. So content, is it accurate? Is it interesting? Clarity, could I go home and tell my 14-year-old about it tonight and remember, everything?
You did all get your 14-year-old before you came into
He came last year, but he couldn't make it this time. It was a bit rude last year.
Right,
so.
and charisma?
And charisma, which is harder to describe, I guess, but obviously you embody it, Quentin.
Yeah, thank you. I like that. The bribing has worked well. And also, remember, finally, all three of you, I mean, all these 7 are proven FameLabbers. They've done it before, so what's going to make the difference? What are you be looking out for? They're all going to have content, they're all going to have clarity, they're all going to have charisma. What's going to edge you towards a winner? What are you going to be looking out for, Anna?
I think I'm very, very keen in learning new things tonight and in learning them in a very FameLabby way, so that's what I will be looking forward to.
I think I'm going to be disappointed when we get to 3 minutes and they have to stop, and I want to hear more because I'm gripped by what they're saying.
All right, so ones who kind of make the page turners, effectively.
Yeah, page turners, yeah.
Right.
I think what really works for me is if it's a story and if it's told from the heart. That's the most important thing.
Okay, that's simple. Same. You've all got 30 seconds to try and incorporate that into your presentation as well. Okay, can we please also thank our judges, because it is a tough task what they've got to do as well. And we have one final cherry on the icing of our not entirely metaphorical cake of tonight, is that after our 7 samurai have shown off their ninja skills, if that's not a mixed Japanese martial arts metaphor, the judges will have a few minutes to make their minds up. And while that happens, we are going to be joined by oven-loving engineer and bake-off finalist Andrew Smith, who has baked us a special ocean-themed Hall of FameLab 10th anniversary cake. And he assures me that is a hell of a brief, a 10th anniversary ocean-themed Hall of Fame. He has promised it is not a fish cake. That is all I know about it. So, right, cakes to come later, and just to avoid any confusion or lawsuits, we are not promising there will be a slice for everyone. But first, we have the main course, a smorgasbord, only without the board bit. And very first, out of our metaphorical hat, we have the runner-up from this year's FameLab Ireland, Joanne Duffy. Joanne is an Irish Research Council postgraduate scholar at NUI, the National University of Ireland in Galway. By day, she's involved in bare-knuckle barnacle research, working on the recombinant expression of barnacle proteins. Now, that would be enough for most people, but no, no, no. By night, she writes comedy science songs for Bright Club. And by whenever it's on, she also works on a weekly science show on the radio with the wonderful title of Schrodinger's Chat. Top title, that. That is on Galway's student radio station Flett FM. That's 101.3 on the dial if you're ever in the west of Ireland. Tonight, though, she is narrowcasting live and direct to the Natural History Museum in London, and the 200 or so countries are out there. So please welcome, at the top of our barnacle bill from Ireland, Joanne Duffy.
My name is Joanne, and I'm a kleptomaniac. I steal genes, not like genes, your genes. A gene is a biological instruction. It's kind of like a computer code written in DNA that tells a cell to make a particular protein. Now, plants and animals make all kinds of really interesting proteins, things like antibiotics and anti-cancer compounds, and even just tasty food ingredients. But if a scientist wants to harvest a large quantity of one of those things, and a plant needs to be, say, 30 feet tall and in its adult form to produce it, scientists run into this mammoth task of having to manage crops and look after test fields and monitor for environmental damage. Which brings me to barnacles. Barnacles produce a really interesting glue that allows them to stick to just about any kind of surface you can imagine. They stick to boats, and they stick to submarines, and they stick to underwater sensors, and they stick to buoys. It's just a sticky situation. So if you want to make a large quantity of barnacle glue, you run into environmental problems when you try to culture them in the open ocean. So I steal their genes. I take the gene that makes barnacle glue, and only barnacle glue, and I insert it into a bacterial species. There are more bacteria on your tongue right now than there are people on Earth. Sorry to make you uncomfortable, but my point is that they're tiny. So tiny that I can fit billions and billions of them in a flask this size. Which means if I genetically engineer them by inserting the gene I've stolen from the barnacles, I now have billions upon billions of cells all making barnacle glue without me having to go outside at all. We want to use this glue to glue wounds closed and to cement medical devices in place. And because it works underwater, we hope that it'll work in your blood. Somebody once said that evolution is a great tinkerer, but the truth is that scientists tinker even more. This process might sound a bit like science fiction, but it's actually well established, and it's used to make all sorts of products that touch your daily lives, like the MSG salt that's put in your Chinese food to make it so tasty. That's made with bacteria. And the insulin that your diabetic friends use to keep themselves alive is produced by stealing the human gene for insulin and inserting it into a yeast species. So I'm a kleptomaniac. I steal and use genes from organisms that I've sometimes never seen, and for now, I'm sticking with glue.
Great, thanks. So you say that you want to make this barnacle glue. Has it been used before in humans? Is there anything known about?
So it's been used in mammal models and in the basically rats, which means that we have a rough idea of what it does in the immune system, and it doesn't cause an immunogenic response, which is just a fancy way of saying it doesn't cause nasty allergic reactions. So the mammal immune system doesn't respond to it as though it's a foreign thing that it needs to eliminate. So that's very promising, because that's the first thing that we look for when we're going to put something in somebody's body. If their immune system is going to attack it, it probably shouldn't be in there.
And has it got any other uses outside medical side?
Yes, so adhesives is actually a huge billion-dollar industry, which I didn't know until last year. So adhesives are really important for just about anything you can imagine. And what's unique about this, obviously, is the fact that it works underwater. And it's not only underwater. Bacteria are what we call an intertidal species. So if they are here stuck to a rock, the tide comes up over them and recedes back out repeatedly. And obviously the water has a high salt content, so it's extremely hardy stuff. It's actually very, very difficult to degrade it, which is another aspect of the research, looking for ways to actually get rid of them, because they stick to boats and sensors and so on.
Is there an anti-glue that you can use to unstick your fingers when you get this stuff on them?
So they have an anti-fouling system. The ingredients of it are actually quite secret. So the US Navy have a particular compound that they put in the paint that they paint their Navy ships with to try and deter barnacles from sticking to them. But it works, but only in a very limited way. And barnacles very quickly evolve ways around the anti-fouling compounds.
You don't want to make a mistake with it then?
No.
Okay.
Time for one more quick one, if you've got one.
I'm not sure if it's quick, but I'm very curious about it.
Yeah.
So how easy it is to be a gene kleptomaniac? How do you steal the genes and put them in the bacteria?
So
That is not easy.
No, it's not. There's a few different ways, but there's a really nice technique called, and stick with me, fluorescence in situ hybridization, which is effectively where you look for a gene and see if the right bit of an organism lights up. So for example, if I'm looking for a gene for your eye, you can use a particular technique that if it lights up in the right spot, you know that's probably the correct gene. So it's a case of narrowing it down and trying to find specifically the sequence you're looking for, but it takes about 12 months to get to that stage.
Brilliant done the last time. Barely believable barnacle banter from our first FameLab. I'll give it to the second, I think. Joanne Duffy. More bacteria on your tongue than there are people on the planet. Now, that is a classic pub fact right there. Right, having started with B for barnacles, we're staying with bees, although still on the FameLab A-list, because we've got this year's runner-up from Bulgaria. And he is Veselin Rusinov, but it's not just B for Bulgaria, it's also B for Batman. Veselin is a computer scientist who's been teaching kids and students robotics on and off since he was 18. The off bit was when he went off to Austria for 6 years to work as a software engineer. He also fixes cars, makes websites, likes reading, but all the time is guided by his maxim for life, which is always be yourself. Unless you can be Batman, then always be Batman. So please welcome the second of our 7 wonders of the FameLab world, Veselin!
Hi. When I was little, I really wanted to learn how to swim, and I asked my father how to do this. So he said, okay, I'll teach you. And he used to hold me under my belly, and he said, move your arms and legs, and I'll move you around. Then I grew a little, and they bought me a live tube. And my father said, again, now move your legs and arms, but have in mind that now you're choosing the direction. Some years later, I really learned how to swim, and I had to synchronize my arms and legs movement, and I had to think where to breathe in and to breathe out. So this is more or less how robotics developed. In the beginning, it was only move motor A, move motor B. Then there was introduced the if-else statement, which was basically move motor A, if you see blue, move motor B, else motor C. Then this was not so good, and people wanted to do really complicated things, and the if-else statements were no good. So they developed really hard algorithms. One of them is neural networks. Neural networks really tries to copy the network in our brains. So let's take this robot. In the middle, we have the neuron, which here is represented as a controller. And inside of it, we have a so-called activation function. This activation function is responsible for the input, for the output based on the inputs. For input, or the so-called dendrites, we have the ultrasonic sensor. And for the output, or the axon, we have the motors and the speakers of the robot. So now when this robot sees something, it decides to scream and to run back. So now imagine that we have tens of thousands of these robots and they are connected. So this is our neural network. First, we have to teach it. Let's say we are making a neural network that recognizes dolphins from octopus. We take 50 images of octopus and we start showing to this is octopus, this is octopus, and this is octopus, and this is octopus. Then we change to 50 images of dolphins and we start, this is dolphin, this is dolphin, this is dolphin. And then we have it trained. Then we have to test it. We test it with different images of octopus and we mark which is correct and which not. If we are happy, we continue. If else, we tweak a little bit the activation function. With neural networks, in the future, we could potentially make robots that could scavenge the whole oceans looking for treasures and even find the Atlantis. Thank you.
So Veselin, your robots, are they going to be looking for life in the oceans, for the seabed, or do you see them as being sort of roaming the oceans to collect lots of different types of data?
This is just a potential idea. It's not, but it could, neural networks are used to analyze big data. So they could collect all this data about the oceans, what's in there, and based on some calculations and of course some analysis on the structure, they could potentially find if there are some lost treasures or artifacts or the Atlantis, as I said.
Okay.
So it is said that with robots taking up some jobs, then the human mistake is going to be eliminated. And I kept wondering when you were saying that we're teaching them to separate the octopus from the dolphin, are we ever going to have, a neurotic network so good that they're going to be flawless and just go on, or are they going to mess up with octopus and dolphins?
It really depends, because it depends on how we teach them. If we show them similar images of octopus and similar images of dolphins, they'll be able to recognize only these, and everything that is really different from them. For example, I don't know, octopus with one less leg, it won't be recognizable for them. So this really depends on us, who we teach the robot, and we that give the data to the neural network. So it could be flawless, but it depends. Yeah, really depends.
So robots aren't taking over the world just yet? Is that what you're saying?
Yeah, no, not just yet.
So the scientists are still safe. I'm glad to hear that.
I hope so.
I'll have to leave it there. Neurons plus robotics, but not neurotics. from the real VR of Veselin Rusinov. And yes, he knows Veselin sounds like Vaseline. We've explained that already. I was fascinated about how it can separate octopuses and dolphins. I didn't even know they got into fights. Right. Third up, we have our Greek-bearing gifts, 2017 FameLab Greece finalist George Petralis. It might be a nice little bit of nominative determinism, but having a surname that starts with Pet, George is a vet, but he has a particular interest in healthcare for wild animals, although he's also got a dog and a horse, both of which I'm presuming are reasonably tame. His poster on educating vet students won first prize at the Animal Welfare Conference in Utrecht, the Netherlands, and his presentation tonight could do the same. Get ready for Greek geek speak from FameLab veteran and FameLab vet, George Petralis.
So, since I was a little kid, I was amazed by the creatures of the ocean, but especially whales. Back then, I had the stupid idea that I could have a whale pet, but first, I had to find one and convince it to come with me. Well, I had to learn to speak to whales. But then what I found out is that whales don't just speak to each other, they sing to each other. And they do that with a special structure they have in their head called the phonic lips. The phonic lips, exactly like the human vocal cords, produce sound when air passes through. But the fun fact is that the phonic lips are situated in the nasal cavity, so it's like singing from the nose, just like Britney Spears does. Well, not exactly, because if you sing to a whale, "Hit me, baby, one more time," she won't respond with, "Oops, I did it again." Because, you know, whales don't use words, they use other sounds like grunts, groans, moans, even snorts. Another difference between Britney Spears and the whales is that the whales have a very low voice, so if they were opera singers, they would be baritones and basses. Well, do you remember Dory from Finding Nemo? When she spoke, she was like, "Hello." Well, she got that right, but not the accent, so... You would ask, why would whales sing? Well, remember that eyesight underwater isn't very helpful because there's not much light. On the other hand, sound travels 4 times faster in the saltwater than it does in the air. So there you got it. Sound underwater is the major mean of communication and interaction with the environment. So whales actually sing for pretty much every aspect of their life, for finding a mate, for finding food, for finding the way, for chatting. You know, man mimics the nature, so they do exactly the same thing underwater. The problem is that man-made communication sounds underwater are very loud, causing a phenomenon called the noise pollution, which is that they alter the natural behavior of the whales because the sound is very loud. So you've experienced noise pollution too. If you go to a middle of a playground with children shouting all around you, what would you do? You'd probably want to get away. So that's what whales do. They try to get away from the sound, from the source of the sound, as soon as they can. So they rise to the surface much faster than they would normally do. The problem is that the change of the pressure causes temporary or permanent deafness, eyes and ear bleedings, or even strandings. So the scientists are trying to create some sonic-excluded zones where whales can live happy. So, after all this, I went to my mom and said, "Mom, I want to adopt a whale and save it from all that." But she said, "No, no, you can't." So I got a cat instead, and thank God, because a whale can fit in my room.
It was very interesting to find out that we are actually doing something to protect them, like having these exclusive zones. But I was wondering, because you said whales are like baritones, so what if humans started having frequencies more like to a tenor or a soprano? Would that solve the problem?
No, because the problem is that the sound is very loud, not the frequency. I mean, it doesn't matter if I sing like or he. The difference would be if I would be very loud, like shouting. So it's not how high or low the voice is, but how loud.
I see.
Just out of interest, do you know what the earliest recording of sound under the sea is from? So do we know what the sea sounded like?
Yes, we
80 years ago?
we have recordings from different periods of time, and we see that the whale's song is changing towards the year because of the man interference underwater.
So you mentioned sonar. Are there other sources of noise pollution from us, from our activities in the ocean?
Well, I believe the sonars are the major, but I think that everything that we do underwater and produces a sound, like some bomb experiments, can have this effect underwater.
Okay.
Thank you.
Very interesting.
Okay. He's from Greece. He's not from Wales. He is George Petralis. Judges, you've got 2.5 minutes for questions. You've got more time than you think, you know. You're doing... I've rushed you too much. That's what I do. I like scaring judges. It's one of my jobs. Okay. So if nothing else, this has been an educational evening. You can now tell the difference between a whale and Britney Spears. That is your take-home fact. Now, we are now slap bang in the middle of our 7 scintillating science showcases, and Piggy in the middle is 2017 UK FameLab finalist, Dr. Mahasweta Saha. A research fellow at Essex University, Mahasweta is a marine chemical ecologist who loves eavesdropping on how undersea plants and animals communicate with each other, and she's doing that in an attempt to understand what their conversation means in terms of global change. Now, Mahasweta is big, huge, ginormous on taking science out of the labs and engaging with the public and underwater creatures, whichever happens to be available first. She says science communication is a hobby, and she never misses a chance to talk to schools, to take part in university events, or even in exceptional circumstances, to distill all that knowledge and put everything into just 3 minutes of a FameLab presentation. So, make it loud, make it proud for our UK entrance in Hall of FameLab, Dr. Mahasweta Saha. I'm sorry.
Hello, Nemo. Wie geht es dir? Nemo? Nemo? Ah, in the movie, he spoke English. So let me try English. How are you doing, Nemo? 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 Doesn't respond. Very unpolite. What's wrong with this guy? Ah, my bad. Nemo doesn't speak German, English, French, or Spanish, but he communicates. His language is completely different from us. It's called infochemicals, information-carrying chemicals. Chemicals are called the language of life in the sea. They are used to find food, find a partner, find home, and manage their day-to-day life. Not just Nemo, but even his friends uses infochemicals. 50% of the oxygen you breathe comes from the oceans, produced by the phytoplanktons. They can run away from their enemies by sensing their chemicals, or they can even find the love of their life by sensing their pheromones. Chemical communication can be so strong that if you treat this ball with the correct female crab pheromone, the male crab will try to make love with this ball. Poor guy. So you see, survival and having a family is so much dependent on chemical communication. Now with climate change, this communication is getting disrupted. Ocean acidification, which is a result of high carbon dioxide in the atmosphere. With that, Nemo babies and even babies of Nemo's friends, they cannot detect their right home, or they cannot detect the smell of their predators. Gets eaten, which means reduced population. As a result, reduced marine biodiversity. this is not just a problem of Nemo and his friends, but it's also our problem because we are connected to the oceans in every possible way, from the water we drink to the air we breathe. So let's protect our oceans, else Nemo and his friends will be lost forever. Thank you.
Thank you. So you talked about the pheromones.
Yeah.
Are they specific to the species, or is there a regional difference? I mean,
Yes, they are specific to specific phytoplanktons. So there are different chemical compounds, and yes, it depends on the genus level and also the species level.
Because I heard something about regional accents in cod at a certain stage. I'm just
Sorry,
There was a story a while back about cod having regional accents, but that's nothing to do with the pheromones, right?
No.
Sorry, bit of a curveball there. Don't worry about that.
Can you taste where your cod and chips comes from? So what sort of distances do these pheromones act on? So will Nemo detect something that's meters away or kilometers away?
They need to be a bit closer to their natal reef before they can start sensing the chemicals, and it also depends on the polarity level. So if they are very highly polar, they would travel long distances. And yes, for Nemo, he needs to be a bit close.
So is this similar to sort of sharks picking up the smell of blood? That sort of, I don't know whether it's a myth or whether it's true, great distances, similar sorts of things.
I mean, they obviously pick up the sense. Otherwise, how would they? I mean, this communication can really go long distance. As an example, I mean, the albatross, which travel thousands of kilometers, and they can sense a particular compound, which is dimethyl sulfide, from hundreds of kilometers, and they know the right area to hit. And that's the area that will be full of tasty fishes. So they can go a lot of distances. And also, as I said, it depends on the chemical nature of these compounds.
I know we probably have a million marvelous ways of disturbing the balance in the coral reefs and in the oceans, but I'm always very interested to hear what we can do as citizens to prevent this and how can we help.
Of course, carbon dioxide reduction, the most important point would be we have to reduce these emissions, and on a personal level also we have to take care of these things. And like, for example, if I now talk about the plastic pollution, we can reduce on a personal level, like reducing the waste we produce, refusing straws. I personally do that when I go to a club, or I say I don't need the straw, I don't need the cup, or like checking for products which doesn't have microbeads, and like I think we can do a lot from a personal level. And of course there are beach cleaning activities and many things.
Edging in before this turns into another FameLab presentation, but can we thank for her informed information on infochemicals, Dr. Mahasweta Saha. I don't know what else I kept finding my gaze strangely drawn to the eyes of Nemo while she was talking, just seeing if he changed expression. And Simon Boxall, if I was a BBC News crew, I don't like this thing about, is it a myth or not, about sharks going over. Come on, I want a definitive answer from you. You're the expert on everything to do with the ocean.
They can pick it up from some distance, but there's a bit of a myth about sharks attacking people. And in fact, the University of Miami have this wonderful record of all shark attacks that have taken place over the last 200 years. And there have been 5 deaths in the UK because of shark attack. I rephrase that, attributed to sharks. 3 were off the northwest coast, where they were spotting basking sharks, and unfortunately this basking shark collided with the ship, and the boat turned over, and the 3 people drowned. The best one...
It's not really a shark attack.
Not really a shark attack. The best one was a blue shark off Cornwall, and 2 people were killed in the 1950s because they were trying to dynamite the shark and blew themselves up instead. So actually, they were recorded as shark attacks, but sharks aren't that dangerous, really.
Right.
Shark defense, yeah.
Next thing you'll be telling me the Sharknado films aren't real. Right, okay, well, Simon, thank you. Now, anti-penultimately, we have our Spanish FameLabber, Andrea Campos Candela. Now, you remember at the start I mentioned about kings and queens and presidents. Well, Andrea took part in last year's Spanish final, which, like most years, actively involved Queen Letizia of Spain. King often goes along too. And this year, the president of Mauritius was one of the judges of the FameLab Mauritius final, where I had the unfortunate job of being the host. So that's a tough gig that was. And in case you're wondering, NASA, when they held the first of their finals, they got the original Lieutenant Uhura, Nichelle Nichols, who fact fans is in the current Sharknado film, 2017, to host their final as well. See, it wasn't just wafflage, unlike the rest of what I say. Anyway, back to Andrea Campos Candela. Andrea is a PhD student in marine sciences and applied biology, exploring the role of behavior in ecological processes and looking at how the different personalities of fish affect their behavior. This is different from Claire's thing about fish having regional accents. So halibut, very grouchy. Dover sole, incredibly vain. Who knew? Maybe not. Appropriately for somebody immersed in marine life and adept at finding stories in the sea, Andrea Campos Candela is an anagram of ocean landscape drama. So prepare for drama and oceans, but probably not landscape, from our FameLab Spain finalist, Andrea Campos Candela.
At the school, I was really shy. I used to hide, trying to go unnoticed. Obviously, that had its consequences on my social life, less interaction with people, less reproductive success. But anyway, I managed to grow up, and today I study the personality of fish. Like humans, fish have consistent personalities during their lives. There are fish more or less bold, aggressive, explorative, active, and social. Let's see. Fish move in the space randomly, and thanks to an attractive force, they remain around a point that is called the center of the home range, their house. Risky fish goes farther away from their houses, while others barely keep out of them. The more space a fish explores, and the bigger the home range is, the more food will encounter, but the more predators as well. So, to move or not to move, that is the question. Whether it's safer in life to hide from predators and risks of an unforeseen fortune, or to take arms against a sea of fear and by opposing overcome them, to eat, to live. Bold fish have this second strategy. They are more active and riskier. That leads them to encounter more food and grow faster. With bigger size, they will be able to avoid predators, and soon they become bigger mothers. And here we have the base of life syndrome. Life strategies to grow fast or to grow slow are correlated with certain personality traits, to be bold or to be shy, and they co-evolve together. But what would happen if conditions change? Imagine we are in the bottom of the sea. The light of the sunrise coming through the water. Oh, small pieces of juicy aperitifs dangling over the seas of leaf grasses. There, for anyone, bold fish rush to take the food, but suddenly they feel a sting in the mouth and a mysterious force drags them up. One, another one, they disappear. Once anglers and fishermen live with their hooks and nets. Only shy fish, those that looked on with fear, remain. Hence, personality traits have changed, and fisheries are inducing evolution in the behavior of fish. To move or not to move, that was the question. Whether it's safer in life to hide from hooks and nets of an unforeseen fortune, or to take arms against a sea of troubles, and by opposing, suffer them, to die, to sleep.
Andrea, do the fish learn? Do they change their characteristics as we change our habits? So do they suddenly become shy?
The fish, you mean?
Yeah.
So actually, there are fish that are shy and there are fish that are bold. So they already have personality traits, which means that they have behaviors that with time we see that they repeat this behavior. So yeah, if I understand your question, fisheries are inducing shyness, you mean?
Do they evolve? Do they evolve with this?
Yeah.
Do they change their braveness in
Yeah.
light of what
So
we do?
what's happening is that survivors are those that are going to have offspring. So if there are some personality traits that are heritable and survivors are shy, so are those fish that are more active, more riskier, so are those that are closer to their center of their home range. So the future generations will be like this. And yeah, so there is a lot of research now studying how the behavior is changing in the populations, maybe also through this fisheries-induced selection, because maybe behavior have a stronger role than we were thinking about before.
So does that mean that they don't change their behaviour within their lifetime? It's only when it's passed on? I mean, are they ever led astray by friends who are a bit more risky than them and kind of take them out?
So behavior is really, there are a lot of plasticity in the behavior. That makes also that the behavior is really difficult to study. It happens like us. So we learn, for instance, also fish and other animals learn, and they can, of course, change the behavior during their life. But there is something that is like the base, that is what is defining the personality. So personality is not only behavior, it's the part of the behavior that it repeats again and again. So it's the part of the behavior that can be irradiated. So yeah, behavior is really plastic, and that is what makes it really, really difficult. It's like our hair. We can cut the hair. It doesn't mean that our future generation is going to be with really short hair. So behaviors happen something like this. So
You've got time for a tiddler.
I have one. So, okay, so fish can be shy or bold or inquisitive and explorative. Can they have even more sophisticated personality traits? Can they be sentimental, romantic?
That's could be, but I'm not so. If I answer this question, I will be giving my opinion, not a scientific knowledge. That's really subjective. So if we can describe in an objective way what it means to be romantic, maybe I can answer it. But we need an objective way to describe those kind of feelings.
I think it's a brilliant answer to say I can't answer that because it wouldn't be scientific to start speculating. So can we please, again, time for our Spanish Tavares, Andrea Campos Candela. And I like Claire's thing about kind of, can fish be led astray by other fish? It's the kind of Ben Stokes underwater defense, I think is great as well.
It's on my mind.
And in case, by the way, anybody was struggling to place that to move or not to move quote, Andrea, it was, of course, from Shakespeare's great underwater tragedy, Halibut. We are full Fathom 5 FameLabbers down and 2 to go. And last but one is our 2017 Netherlands finalist, Selma van Staveren. After an MSc in molecular medicine, Selma became a medical doctor in 2014 and is now doing her PhD in immunology, all of which might seem impressive, but absolutely nothing to do with the oceans. Selma, though, has an interest in how what we do on land has an effect on the waters around us, which in turn can have a very unhealthy impact on humans and lots of other creatures. I could say more, but it's rather better if she does. Representing the Netherlands, please welcome Selma van Staveren.
I would like to start with making a toast to our health, because that's what matters most. But it might be in danger, and I will tell you why. The plastic pollution in our oceans is increasing. And like a sugar cube slowly dissolves in a cup of tea, the plastics in our oceans also gradually break down into tiny particles. These tiny particles are called microplastics. Microplastics are so tiny we cannot see them with our naked eye. We found them in fish, mussels, and in oysters, but even in salt, honey, and in beer. So microplastics are harmful to marine life. But what about the effects on our life? Would it be harmful for me to drink this beer? Let's follow the plastics in this beer. So the beer reaches my stomach and passes through my intestines. Now scientists have shown that the microplastics can pass through the intestine wall. Which cell type in my body would be the first to encounter a microplastic? We all have a cell type in our blood that scans our body for intruders. These cells are called neutrophils, and they are known because they are very important to keep us healthy and to prevent us from infections. The neutrophils actually eat bacteria and then kill them. Now, microplastics are also intruders to our body. So, would the neutrophil be able to find the microplastic? And would it eat and kill it? Or, is that even possible? What we did in the lab is we added a microplastic to human blood. And in the blood there are the neutrophils. And what we saw is that the neutrophils eat the microplastics, just like they eat bacteria before killing them. Now, a neutrophil is not equipped to degrade a plastic particle. So we wondered what would happen to the bacterial killing capacity of the neutrophil. We saw that neutrophils with microplastics are way less capable of killing bacteria. This is quite scary, because if our defense system is down, we can get sick. Now, of course, these experiments in the lab are not like real life. So we still want to find out whether this also happens in a living organism. Our next experiment will be to feed a mouse microplastics in its drinking water and to find out where these microplastics end up. Do the neutrophils find them? And are they less capable of killing bacteria? By this, we hope to find out whether it's right to toast to our health with a nice cold beer.
I keep wondering if the microplastics are big enough so that we can see them. Is there any way we can filter them?
Yeah, so there are companies that try that are working on that to filter the microplastics. Actually, you have a big company in the Netherlands that's just starting up that want to filter the plastic soup in our oceans. But then we also reach to this problem that for bigger plastics, this will work well. But the smaller we get, and actually we don't know how big the problem is, because the real small ones we cannot even see on our microscope. So they are transparent, so it's really difficult to find them. So I think the problem is so big, and I think we don't know that much about it actually. Yeah.
And is the problem worse in beer, or was that just an excuse to drink on stage?
I like beer. No, it's not worse in beer. Now I think it's worse in mussels and oysters and organisms that really filter the ocean water the whole day. But on a side note, they did also find it in beer. Yeah.
Is there a danger if we filter the oceans? Do we take all the good stuff out as well? So if you're looking at sort of some huge filter to take all the microplastics out, what about the plankton, the phytoplankton?
Yeah, that would be also a big problem. Yeah. So these are things that companies are thinking about all the time, and also governmental organizations. But there's not really a solution that probably will solve the whole problem yet. But I hope it will come. Yeah.
If we cannot filter them, is there any way we can support our immune system to fight them?
Yeah, that would be also an interesting thing. There is also some knowledge about bacteria that can degrade plastic particles, but until now, I'm not sure about the real. But they say it still takes 7 years to degrade the whole plastic bag. So, but they're also in these things to find organisms or cells that can degrade the particles or find it and catch it. That would be also a nice solution, but it's not that far yet.
It can take just a couple of minutes to degrade a FameLab if they get the wrong kind of questions. So can we please thank Drastic Plastic Fantastic from Selma van Stafferen. And Claire, you picked up on Selma following in the grand tradition of scientific self-experimentation by drinking beer on stage. But imagine she comes on, she starts eating oysters. You're going to lose the popular vote if you do that. And I did mention popular vote. There will be an opportunity for you to vote in a second very soon, because last but statistically unlikely to be least, we have FameLab Poland 2014 champion Joanna Bagniewska. I should say Dr. Joanna Bagniewska. Joanna is a zoologist now based in the UK as a teaching fellow at Reading University, but she's also, like Maha Sweater and probably all the other FameLabbers and probably half the people in this room, a devotee of science communication in all its forms. Besides FameLab, she's won I'm a Scientist, Get Me Out of Here, given a TED Talk, debated Can Science Save Football? She's a Liverpool fan, like my wife. Hello, my wife. And performed science at stand-up events. But how deep is her knowledge of the oceans? And which bit of it is she going to sail us through in the next 3 minutes? Go mildly wild for your final FameLabber of the evening, Dr. Joanna Bagniewska.
Open wide. You can say this to a hungry baby, but can you also say it to a hungry penguin? Well, kind of. But you'd have to be somewhere in the ocean and stick out a nice yummy fish. Now, for scientists who are interested in the feeding behavior of aquatic animals, but who don't want to get themselves wet, there's a different method, and that is data loggers. These are small devices that are stuck to free-swimming animals that record all sorts of data from sensors like temperature, depth, or magnetic field. Okay, but how would that tell us just how peckish a penguin is? Well, we can use Hall sensors. Hall sensors are small devices that measure a distance between a magnet and themselves. So what we do is put a magnet on one side of a penguin's beak and the Hall sensor on the other, and voila. You know just how big a fish a penguin is going for. The bigger the angle between the two parts of the beak, the bigger the fish. Okay, but like babies, penguins sometimes open their mouths for things other than food. How will we know that our Hall sensors are not deceiving us? Well, we could put another Hall sensor on the other side of the penguin and see what comes out. Or we could put another sensor in the penguin's esophagus. Now, don't try it with babies at home. But with the penguin's esophagus, you'd put a temperature sensor there. And with a warm-blooded penguin eating a cold fish, you would notice a drop in the temperature. And so, hey presto, when you retrieve your penguin, you're able to record or to read all the data from the data logger and know how many fish it has eaten and how big they were. Okay, but why would we do that? Why are we interested in what penguins eat? Well, simply because we want to know what the prey availability is for them. So if we notice that our penguins consistently eat smaller and smaller fish, that might mean that there just aren't any large fish left for them to eat. And that could be an indicator of something going very badly wrong in the ocean's environment. And so, just like that, a penguin's beak is a nice indicator to show us whether the outlook of the ocean is a little bit, well, fishy.
So how easy is it to persuade a penguin to be instrumented up?
Well, it's not done on a volunteer basis, put it this way.
So They don't sort of sign up and sort of get a free beer, for example.
No. No, the data loggers are glued to them under anesthesia. So it doesn't hurt them, nothing bad happens with them, but they don't exactly get a choice.
Okay.
And how do you get them back?
Well, okay, this is a question that I haven't personally been involved in because I've worked on data loggers, but with mink. So slightly different. We don't put the whole sensors on their mouths. We use data loggers for something else. But with penguins, when they go on land, you can catch them. They are not exactly very fast.
I'm a little reassured that you said before that it is safe for them and we're not hurting them, although I would be very mad if somebody put a data logger in my esophagus, to be fair. But why did this research first start? So did we have any indication that something was going wrong with their nutrition?
I think, well, I don't know how this particular research started, but I think, like with a lot of things, you see different opportunities, and it's a accumulation of different circumstances. So you notice that the oceans are not doing that well, and at the same time, over the last few years, data loggers have been miniaturized, and so from something that has been a few kilograms big, you now have moved on to a teeny little device, and people think, oh, this is really awesome. I know what we can do with this. We can kill two birds with one stone. We can get to play with awesome data loggers. Yes.
No penguins.
No penguins. No stones, penguins. No, none of that. Metaphorically, and we can find out more about the ocean condition. So I think it's a pretty cool thing to do.
One quick question: do you have to recover the penguins, or do they actually, when at the surface, can they transmit the data via satellite?
It depends on the data logger.
Okay.
So with the data loggers, the biggest downside is the battery life. And so you can have the very basic ones where you have to retrieve the animal, or you can have the super fancy ones that can detach themselves and float to the water surface and then send a satellite signal, and it's all easy from there. But they tend to be a bit bulkier.
Okay, I have to leave it there. Applause, please, for penguin instrumentation from Poland's Joanna Bagniuszka. And apologies if there are any younger people in the audience who are left with disturbing images of penguins being with their beaks held open at strange angles and magnets in there as well. So that was the last of them. They've all been not just good, they've been FameLabulous. It would be nice to leave it there. But even though tonight is an exhibition match and has no impact on their FameLab Champions League rankings, we do need a winner, a king, an emperor, or some other kind of penguin. Judges, you have only slightly longer than a FameLab presentation to go out there and make some decisions. So you have to decide who will get the star prize, the singular honor of carrying home this trophy. Woo, woo. Yeah, it's not easy to get a trophy that small and special like that as well, so okay. Can we please send our judges on our way with some applause, please, because they've got some difficult things to do. Now, while they're away deliberating, we are going to have cake, or at least talk about cake, and we're going to welcome 2016 Bake Off finalist Andrew Smith.
Hello, Quentin.
And Andrew has also very kindly, as well as having a little chat with us, agreed to act as a sort of human clapometer. Now, it's very simple. This is just for
I've not
fun.
been calibrated recently.
Haven't you?
No.
Well, we've got somebody out there. We've got some spare penguin equipment, so if you wish as well. So what we're going to do is I'm going to give you a quick reminder of all 7 you've just seen. Then we're going to have a little burst of applause, well, hopefully a big burst of applause for each of them, and Andrew will judge who the applause is loudest for. So just to remind you, don't go clapping yet. So from Ireland, Joanne Duffy. From Bulgaria, Veselin Rusinov. From Greece, George Petralis. From the UK, Mahasweta Saha. From Spain, Andrea Campos Candela. From the Netherlands, Selma van Staveren. And from Poland, Joanna Bagniuszka. Okay, so now is the bit where you come and make some noise, but proportionately. Try to put aside factors like geographical bias, subject familiarity, gender, unrequited lust. weird feelings towards penguins. Just applaud on the basis of who you think gave the most amazing presentation and dealt best with the judges. And if you're watching the webcast, please, it's very important, clap along as well. We want to keep them involved. Okay, so are you ready? We'll only do this once, so keep it all there. First of all, barnstorming barnacles and bacteria hysteria, Ireland's Joanne Duffy. Robots all at sea from our Bulgarian Batman fan, Veselin Rusinov. Great Greek geek speak, George Petralis. From the UK, but highlighting how Britannia waves the rules, Mahasweta Saha. Our Spaniard in the works, Andrea Campos Candela. Oh. Some bonus whooping there as well. No double Dutch from Netherlands FameLabber, Selma van Staveren. And finally, punchy, pinchy penguin formation from Poland's Joanna Bagniuszka. This is tough. The good news is the prize is of almost no consequence. No, but it's a really lovely, special science book, so they'll be excited by that. They don't get a Victoria Cross. They don't even get a Victoria Sponge. They just get the little prize. Andrew, your decision is binding.
So it's very close.
And I'll give you the list of names in case you need a little reminder.
But I think with a whisker, it's going to be the Netherlands and Selma.
Woo! Congratulations, Selma. And we will give you your prize in a full... So, Andrew, we've just got a few minutes to chat. You know, you've ended up here.
Yes.
But it's like kind of odd route because you went to FameLab UK final back in April, and then you were kind of gently harangued and meringued until you said yes to coming along tonight. Now, were you there because, as far as you're concerned, STEM and baking and science are almost as important to you as being on the TV?
Yes, absolutely. And actually, the FameLab UK final this year was the first time I'd actually encountered FameLab in any way, so I was kind of intrigued. And I went along with Tom from our year's Bake Off as well. And I was just fascinated. I just loved the format. I loved just 3 minutes. No death by PowerPoint, which I work in engineering, and we get a lot of death by PowerPoint. So it was really refreshing. Just having 3 minutes, people being really concise, and yeah, it was something I wanted to be involved with. I've done a little bit of science communication myself, nothing quite as daunting as you FameLabbers. I'm all very impressed by all of you.
Would you ever think about
You know, I would have, and
entering?
I think if I hadn't have done Bake Off, I definitely would have gone for
okay.
it.
So take us through the history. Is it engineering then baking, or baking then engineering, or simultaneously, parallel processing?
It was a bit of both. I like to think they complement each other quite well. When I went in the tent, I baked from quite a young age, but I only really did it a lot more seriously after I left university and had my own kitchen, so I wasn't treading on anyone's toes.
Just a kitchen, or was it a whole flat? I mean,
sorry. Well, at the moment, I kind of have I call it the pantry. It's about a quarter of the living room. My housemates aren't so happy, but it's kind of overflowed from the kitchen. But I kind of took my precision from engineering and like a first principles approach. So many people, when they get into baking, I mean, if you were painting, you wouldn't expect to be Picasso the first time you picked up a paintbrush. So a lot of people, they pick up a recipe book, go for a really complicated recipe. It inevitably goes wrong, and they say, oh, I can't bake. But you've got to start with the building blocks and work your way up, and that's kind of the approach I took, and it seems to fare
Well, I remember
well.
a cake of yours, not exactly building blocks, but pretty close. It was gears. It was the Da Vinci
Yes.
gears. Now, that's a kind of visual metaphor of engineering rather than being an engineered cake. That's like giving like a Man City fan like me a cake that's the shape of a football pitch
Yes,
or whatever.
yeah, exactly.
So, but how much actual engineering do you put inside your structures of cake.
So I guess that was the cogs were
Yeah.
that got my mind turning.
So see
One
what
of
you did
yours
there?
there, Quentin.
Hang on, not every bad pun has to be trademarked as mine, but yeah, fine.
But as you say, I wanted to put more real engineering. I didn't just want it to look like things. I wanted it to show something. So what I do now is I've been doing some more science festivals and going into schools, and I use baking to show engineering principles. So I do something where I go in, I make a sugar bridge out of caramel, I reinforce it with strawberry laces and kind of tell the tale of reinforced concrete, which sounds really dull, but it's actually really interesting. And then I go to honeycomb and how that's used in space structures. So I'm kind of using these materials that we see in the kitchen, which show principles that engineers are able to cleverly apply elsewhere. So it's really engineering and baking rather than just something that looks like engineering.
Because you've used this term baconeering, haven't you?
Yes.
And of course,
which is a real word.
It's a real word now,
Definitely
right?
a real
but
word.
we've obviously got Heston Blumenthal on the field of play with molecular gastronomy, and obviously baconeering is not the same, is it? You've got to be clear about this.
No, yeah, a lot of people are doing, and rightly so, I find it really fascinating what Heston's doing, molecular gastronomy and why things happen in cooking the way they do, the chemistry and physics behind it. But I guess an engineer's job is to take science and kind of make it useful for humanity, have that kind of inventive step. And that's what I want to celebrate, because I don't think it's really being done at the moment. And baking and food is really universal. It's something that everybody understands. So I kind of lure people in with the baking and then hope that they stay for the engineering.
I was going to ask, because obviously we know you through Bake Off, but are you just a baker or are you a full spectrum cook? Are you also applying engineering to doing the Christmas dinner as well?
Ooh, well, I love a spreadsheet. Anybody that saw the final last year knows I love a good spreadsheet. And Christmas dinner this year, I think I have the spreadsheet pretty much down for Christmas dinner. And I did have an idea for a cookbook, actually. Would people buy kind of spreadsheet bakes, something that showed you where you were? Okay, no one's keen for that. No one's keen. Yeah. Okay, that was an awful idea.
It's even worse because you've just heard how loud the crowd can be, and you just go like,
I know, yeah. Spreadsheet bakes, yeah, tensive enthusiasm. But I think bringing engineering into the kitchen is a good thing. And people think it's a bit crazy. They don't think it makes sense. But actually, I think there's quite a few overlaps.
How do you manage your work-life balance? Because you're still at Rolls-Royce, aren't you?
Yes,
as I understand these things as well. And so, and what do they kind of let you escape a couple of days a week, or do you bake during your lunch hour?
So I managed to haggle myself down to 4 days a week. I only get paid for 4 days a week. I wasn't just like, go on, give me one free. But I've redone my
I don't think Get Me One Free has ever worked with Rolls-Royce.
Yeah. But that enables me to kind of have one day where I can do the science and engineering stuff and baking as well that I love, but I love my day job as well. I work as an aerospace engineer. At the moment, I work in future aircraft development, so kind of 20 to 30 years' time, more electric aircraft, things like boundary layer ingestion, if you've ever heard of it. So I find that far too interesting to go into baking full-time, so at the moment, that's
boundary layer ingestion?
Boundary layer ingestion,
That does sound like it could be talking about the cake still.
Yeah, so, well, a very brief aside.
It's like I ate the middle of the cake, and that was the bit I shouldn't have ingested first, so you've crossed the boundary layer here, right?
I'll try not to go, I haven't done like a FameLab pitch on this, but it's effectively, it's a lot more efficient to accelerate a lot of air by a little bit rather than a small amount of air by a lot. So what it does is it takes the slower speed air that slows down due to friction over the fuselage and then tries to accelerate that out the back, so it reduces drag and improves aircraft efficiency.
And I guess you've probably been asked this before, but what do you consider to be the Rolls-Royce of cakes?
Oh, the Rolls-Royce cakes. Well, I did actually, we haven't planned this, I did actually make a jet engine cake last year,
right.
which had an RR logo on it, so I guess technically that might have been the Rolls-Royce cakes.
Right, your own cake is the Rolls-Royce.
Sorry, yes,
cakes.
oh yeah, I know, but
okay.
I think if you're asking what my favourite cake was, I think just sometimes simple is best. I love a good lemon drizzle.
All right, okay.
Lemon drizzle's the shizzle.
Yeah. Lemon drizzle, I like that. You've got to copyright these catchphrases.
Learning from all your rhyming, I'm taking
as well as doing the jet engine, you were set another challenge, which is to make a 10th anniversary Hall of FameLab ocean-themed cake.
Hopefully no one's been nibbling back here. Yes, so I
was before the reveal, do you want to tell us anything about the thought processes?
I'm not just stealing the banner, it is actually behind here. So, well, I think if I bring it out, it's easier to talk about. I'll bring it over there into the light. So this is my 10th anniversary FameLab cake. And I'm going to take it off the cake stand just to reduce the me becoming a meme and dropping it on the stage.
I was going to say, that must be the most nervous thing about doing these, is am I going to drop it before anybody even gets to see it?
So on one side, I'll come to the swirls in a second. On the other side, it's the easier to cut out of fondant continents. And I thought I wanted to celebrate, FameLab's gone global, so I thought a globe, lots of ocean on there as well, all tying in with evening. And on this side, it was neater in my head, but one thing, I don't know very much about oceans, but one thing that we actually use in engineering is something called the Coriolis effect. And this is what drives a lot of our ocean currents. So in the top kind of 100 meters of the ocean, the shallow part, it's mainly driven by global wind systems. And
isn't it right that it spirals one way in the northern hemisphere and the other way in the southern hemisphere?
So towards the right in the northern hemisphere, towards the left in the southern hemisphere. If the Earth didn't spin,
that would be bad.
Yes, it would be very bad for a number of reasons. But the hot air at the equator, if there was no spinning of the Earth, we would just get air currents, which would kind of just go from pole to pole. But because it spins, because of conservation of angular momentum, when we move away from the equator, we get this kind of turning effect. And that's how we end up with these turning to the right systems in the northern hemisphere and turning to the left. So I tried to recreate that with some swirled icing. So you can come up and have a look, but it is passion fruit Swiss buttercream, so if nothing else, it should be delicious.
Oh. And as a bonus prize for our winner tonight, we are going to let them cut that cake as well. And then I'm going to eat it all. So.
That was always there.
It's about time. The judges are back. But before they come back, we have to thank the amazing, wonderful, bakelicious Andrew Smith.
Thank you very much. Thank you, Quentin. Thank you.
So, who won? Please welcome back your judges. Claire, Simon, Anna. Got a bit more, please. Come on, they've worked hard, they. It is time for 85.714% of our FameLabbers to be disappointed. That is 6/7, maths fans. So, you are officially meant to come to your verdict based on those three criteria of content, clarity, and charisma. I don't know if you took one each. I don't know how you did it, but tell us what you found and who the winner is in whichever order makes most logical, dramatic sense.
So I have to say, when I was bragging before about hiding behind the table of the judges, and I thought it would be so easy, we would enjoy the talks, discuss, and then outside I realized that the hardest part was, trying to decide who the winner was. And frankly, I think we would be still discussing it in a civilized way. But we were told that the cake was revealed. So it felt like a nice time to coming back. But
frankly, we haven't got a winner. We're just here for the cake, and you can all go home.
But what I really want to stress is how much difficult this was for us. And thanks to all of you that made it difficult to us.
Yeah, we should give them a collective round of applause because... that was the lovely last moment of unity and harmony. Now they are about to be ripped apart by these judges.
Well, just to string it out a bit more, we wanted to say something about each of them because we found each of them had, really amazing bits of their story. So Joanne, we loved the concept of kleptomania. I thought that was fantastic, and the little snippets that you put in there, like the more bacteria being on your tongue than people in the world, were really good. Really grabbed us, I think.
And then Veselin, we saw some great stuff from you. This really complex idea of neural networks, and I've had a few PhD students who've looked at neural networks for other things in the oceans. And actually the applications that are out there, and you sort of pinpointed some of those great applications. So I find it fascinating.
And George, we really enjoyed how the variety of the means that you used, how humorous it was, even the singing that you did. That was very original and very nicely done.
And Marsueta, and I get the name that's really hard, sorry, I hope I said that right.
It's not very hard if you're Marsueta.
No.
She practices.
You really touched me. I mean, I felt your passion through what you thought. It really shone through how much you care about the oceans, and that I think is a kind of jewel, I think, really.
And then Andrea, your wonderful shyness, this ecosystem and how the ecosystem's affected, not so much by the, the characteristics, the physical characteristics, but the personalities of the animals in the ocean. And this is a fascinating idea.
I still believe they can be romantic, by the way. Selma,
Help me start a dating agency for marine creatures or something.
I could definitely do. Selma, that was very engaging, and what impressed me mostly was how well you replied to all the questions. Actually, you used the question time, and we learned more things, especially about the immune system and the bacteria, so that was very nice.
And then Joanna, we had this wonderful image of these penguins being monitored day and night with everything they did. I wonder what other senses you could put on them. The mind boggles, but it certainly grabbed the audience. You engage very well with the audience. But we had to make a decision, and it's a very difficult one. So what is a good science communication? It's about engaging your audience, and the winner did that very, very well. They engaged the audience incredibly well. It's about a narrative. It's about storytelling. Science can sometimes come across as being a bit dry. Even in the ocean, it's a bit dry sometimes, albeit wet. But it can come across as being a bit dry. And all of our contestants really brought their science to life, but the winner really brought it to life very, very well. And there's a degree of humour, there's a degree of interest, there's good facts getting across on that humour as well.
I'll just stop you there to say, I still have absolutely no idea which one they've picked, which is great.
Yep. Oh, sorry, Quentin, I thought you were deciding who got it. Now, obviously, we've had Andrew talking to you before we came on, and with the bake-off, as Andrew knows, you had Mel and Sue who had the job of saying who was going home and who was the baker of the week. They still do the same thing apparently on the other channel. Luckily, we don't have anyone going home because basically all our presenters were absolutely stunning and fantastic. So the most difficult job was who was going to sort of go home with this fantastic prize. I hope you've got something to carry it in, because it's got to be, we'll send security guards with you to keep it protected back to your hotel. The winner for this year's FameLab here tonight was Andrea.
Andrea Campos Candela, come on up. Brilliant. Well done. Andrea, congratulations. That must have been tough. We now need the dramatic moment where you put down the trophy, I think, and cut the cake. This can only go spectacularly wrong. I'll try to put the trophy out of the way. There you go. I know. It's a metaphor for the ephemerality of science communication itself. So just get in there.
Officially, the world does end tonight.
I hope that it's not the future of the Earth.
Hold on, I'm getting this. Yeah, okay. Oh, he's realistically created all the rock layers within the Earth as well. It's amazing. Right. Oh, yeah, we didn't think about plates. Not the trophy, Andrew's always prepared. Okay, while the cake is just being put on, because it might be slightly anticlimactically, but dramatically, we're going to have a quick word. Oh! That was like watching clowns at the circus when I was 8 years old, all over again there, that little routine there. We're going to have a quick word from the science advisor of the British Council and the kind of de facto head of FameLab, Adrian Fenton.
Right, I've had an
Applause.
amazing...
He gets applause too, you know.
Oh, well, I can't credit that. I've had an amazing evening, and I really enjoy the opportunity this job offers me to see so much amazing talent around the world regarding science communication that's making science key to the future society. We're talking about the things that science can provide. I want to say a big thanks to our venue and host, the Natural History Museum, to all our presenters and also our judges who have given their time freely as part of this. We've got some gifts to say thanks to all of you for your contributions.
That's
from
all of them, not all of you.
The shops. The cake, we'll stretch it as far as we can, but I hope we've also whet your appetite to find out a little bit more about the activities of the British Council. We have a network, Euraccess, that we work with, where we support research and funding across Europe. We also have FameLab coming up. The rounds are soon. Perhaps some of you will be up here next time. We also have articles from some of the presenters here in our Voices publication, so there are things for all of you, so please do look us up. Many thanks, Quentin, and I hope you enjoy the rest of your evening.
Thank you, Adrian. Yeah, that is it. It only remains for me to echo what Adrian has said. Thank you to our fantastic trio of judges. Thank you to Andrew Smith. Thank you for this amazing looking cake, which I'm going to guard carefully if we need to. I want to thank Adam and Majik for giving us the sound as well. Thank you for choosing to swim in this particular ocean as part of Researchers' Night. Now go off into the wild, uncharted waters of the rest of the evening, and we hope to see you at another FameLab event soon. Have a great evening.